Hi everyone, my name is Monalisa Manhanzva from the University of Cape Town, and the title of my talk is Immunomodulatory Properties of Cervicovaginal Lactobacilli are associated with lactic acid production and bacterial proteome profiles. Just to give a bit of background, South African women are at an exceptionally high risk of HIV acquisition and STIs that cannot be fully explained by behavioral risks, suggesting that other factors are at play, and therefore studies have been carried out to investigate other factors, such as the composition of the female genital tract microbiome. Lactobacillus dominant vaginal microbiomes, which I will be referring to as optimal, are generally associated with the healthy female genital tract due to the ability of lactobacilli to produce protective metabolites, such as lactic acid. This suggests that lactobacilli may be protective against STIs, which leads me to the aim of this project, which was to characterize the immunomodulatory properties of vaginal lactobacillus isolates and to determine the mechanisms underlying these relationships. The samples analyzed for this project were collected as part of a parent cohort, which was known as the Women's Initiative in Sexual Health, or WISH. And the study was carried out in Cape Town and recruited 16 to 22-year-old sexually experienced women who were also HIV uninfected. Participants were followed up from baseline up to 6 months, and demographic and clinical data were also collected. BV was classified using Nugent scores for the parent study. For the current study, we evaluated lactate production and culture acidification by the lactobacilli, which had been isolated from vaginal secretions that were collected during the parent study. We found that lactobacilli obtained from women with optimal microbiota produced more total lactic acid and D-lactate, while there was no significant difference in culture acidification and lactic acid production. We next evaluated the influence of the vaginal lactobacilli on inflammatory responses by stimulating vaginal epithelial cells with lactobacilli cultures, and we measured the levels of cytokines produced in culture supernatants using Luminex. When we compared the cytokine responses, we found that lactobacillus isolates obtained from women with non-optimal microbiota tended to induce greater cytokine responses than those from women with optimal microbiota, suggesting that the vaginal environment may have an influence on the lactobacilli characteristics. To further understand the characteristics of the lactobacilli that may be used to explain the varying inflammatory properties that we observed, mass spectrometry was used to compare overall protein expression between non-inflammatory and relatively inflammatory isolates. We next evaluated the relationships between lactobacilli properties and proteome profiles, and we found that production of D-lactate by lactobacilli isolates correlated positively with D-lactate dehydrogenase relative abundance. We also observed a strong negative correlation between D-lactate dehydrogenase relative abundance and inflammatory cytokine response. In summary, these data show that lactobacillus isolates from women with optimal versus non-optimal microbiota differed functionally, with isolates from women with optimal microbiota producing more total lactic acid and inducing lower cytokine responses. We also found that immunomodulatory properties of clinical lactobacillus isolates are likely multifactorial and may be associated with membrane transport enzymatic activities and lactic acid production. I'd like to thank my supervisors, colleagues, and lab mates, the study participants, as well as the funders who made this work possible. Thank you.